Good afternoon, and welcome to the OSU Center for Clinical and Translational Sciences Inventors Showcase. My name is Rebecca Jackson, and I am the director of the CCTS, a university-wide center that is supported by the NIH National Center for Advancing Translational Science. This workshop series is a collaborative effort between the CCTS and our pediatric partner, Nationwide Children's Hospital, as well as the OSU Offices of Research and Corporate Engagement. The primary mission of the CCTS is in fact simple: to accelerate the translation of scientific discovery to new prevention, diagnostic, and treatment strategies that improve human health. As most of you know, there are many pathways that can be taken to support research translation. Today, our focus is on examining the potential of innovation, commercialization, and entrepreneurship, or ICE, as one viable translational option. Through the power of stories, today we hope to highlight the journey of innovation through the eyes of our local biomedical science inventors as they work to bring innovations to stakeholders and ultimately the marketplace. We hope through their stories that you will gain insight into the challenges that arise and the lessons that have been learned that have led to the ultimate success of these research teams and their ideas. This summer, given the impact of the COVID-19 pandemic that has impacted everything that we do, both in the research and academic activities at Ohio State and across every aspect of our own lives, we are focusing on stories of emerging innovations that have arisen in response to some of the most pressing needs that have been identified within the COVID-19 epidemic. Although some of these projects are still very early in the ICE process, each of these projects do illuminate some common themes. First, the vision of the investigators to recognize opportunities where innovations built upon a foundation of rigorous scientific knowledge and approaches can address a societal or health-related problem. Second, the critical role of team science, which brings people from multiple disciplines with unique knowledge, perspectives, expertise, and skills to work together to facilitate progress on a project that no one investigator could have done and succeeded at alone. And perhaps third, and most importantly, tenacity. The tenacity to move forward and find ways around barriers and challenges that could impede progress. The CCTS, through its partnership between Ohio State and Nationwide, can help to reduce some of these barriers by providing access to resources such as our CCTS voucher program, which helps fund just-in-time access to a wide range of translational science cores across Ohio State and Nationwide, to our grant support of regulatory resources, such as the Drug and Device Development Services at Nationwide, which provides IND and IDE guidance to investigators that you will hear more about today. Our mission is to catalyze and enable your success. For more information on this range of services and resources, including educational initiatives that are available to facilitate translational science in general or specific to COVID-19, we hope that you will reach out to us via our CCT website or visit the COVID-19 websites housed by the OSU Offices of Research or the Infectious Disease Institute. We know that all of us working together can make a difference. It is now my distinct honor to turn this program over to Dr. Christopher Roulette and Dean Lee, who will share with us some of the insights from this project entitled Enhancing Innate Immunity. Starting first will be Dr. Roulette, an assistant professor of pediatrics in the Division of Infectious Diseases at Nationwide Children's Hospital. His clinical and research interests.
Dr. Jackson, thank you so much for that warm welcome. I do appreciate it. My name is Crystal, and I'm one of the infectious disease docs here at Nationwide Children's Hospital and part of the Host Defense Program. And I'm very pleased to present this data, which has been a truly collaborative effort between ourselves and the cell therapy program here at Nationwide, looking for a novel approach, the use of adoptive immunotherapy with NK cells to treat SARS-CoV-2 infection. So to provide a little bit of background of this information, SARS-CoV-2 infection was really first described in December of 2019 in Wuhan, China. And we really had multiple names for this as it was coming out, but as we've learned more, this infection has shown close phylogenetic linkage to that of SARS-CoV-1, a la the original SARS virus that caused the SARS outbreaks in the early 2000s. Importantly, we've seen a wide variation in clinical presentation from asymptomatic illness all the way towards respiratory failure and death. To provide a little bit of context as to where we're at, let's take ourselves back to mid-March when we first engaged in this collaborative effort. And at this time, China was seeing roughly 81,000 cases. They had started to plateau in terms of their total case count, but Italy had seen roughly 35,000 cases. Spain was seeing 14,000 cases, and both of those were rapidly rising as the outbreak extended into Europe. The UK was starting to see some cases, and here in the United States, we had about 9,000 focused mostly in New York City. But what's important to remember about this was the difference in mortality rates. China had reported a mortality rate of roughly between 2 to 3%, although in Italy and Spain, they were seeing mortality rates of 10+%. So what this really led to was this strong charge to find a therapeutic option, what we could do for these patients to try to ameliorate and or prevent those morbid outcomes. A lot of options were suggested early on, and we'll discuss where these are given their importance later on. So lopinavir-ritonavir, a protease inhibitor used mostly in HIV infection, was suggested up front. And that was derived mainly from early in vitro data suggesting activity against SARS-CoV-1 and MERS virus, so seemed like a nice place to start. King et al. Had performed a study looking at this, and first things first is they looked with some in vitro studies. In the top left box, what we have are vero cells that had been the control, so they showed no viral cytopathic effect. Box number B would be those same vero cells that were infected with SARS-CoV-2, but without any agents added. And then most importantly, box D, that was going to be the SARS-CoV-2 infected vero cells, however, were incubated concomitantly with lopinavir-ritonavir at doses that were believed to be achievable therapeutic concentrations in human serum. Importantly, when they looked at viral loads from those cells over time, being at 24 and 48-hour time points, those that had received the lopinavir-ritonavir represented in the orange, blue, and green bars showed significant decreases in viral load, again on in vitro-based data. This led to some initial studies, mostly in case reports. First one here published by Lim out of Korea, where they had administered that drug to a patient and noted clinical improvement in terms of virologic detection as well as clinical improvement of that patient. Hydroxychloroquine and chloroquine were also very early proposed as targets. The mechanism of action is truly unknown. There's been a lot of suggestions being alterations of pH at the cell membrane to disrupt binding, but possibly alterations of viral assembly, transport, and release. When they looked at in vitro data looking at percent inhibition of cellular viral activity, be it with chloroquine noted on the left bar and hydroxychloroquine noted on the right bar, that vero cells that were infected with SARS-CoV-2 when administered with hydroxychloroquine or chloroquine did show EC50s that were rather potent, some as micromolars, which were the target levels that could be attainable in human serum. This then led, of course, to many different studies that have been published. This first one here was published by Chen et al. It was a randomized controlled trial, but it was a non-peer-reviewed study. It had 62 patients in total that were involved, and it had some suggestions of benefit, specifically with reduced symptomatology and reduced pneumonia in those patients. Additionally, a study which would become infamous later on was the concomitant use of hydroxychloroquine and azithromycin for treatment of COVID-19, where the authors suggested that there was virologic as well as clinical benefit, which we'll discuss a little bit later on as we've had emerging data. The last one, which was of importance, was remdesivir. This one is a nucleoside analog that works to inhibit RNA-dependent RNA polymerase. Its most recent notoriety was its application in Ebola patients, though, which was without clinical efficacy. However, there had been prior data looking at its use in other coronaviruses, which was warranting some approach. And amazingly in this, when looking at the EC50 of this drug, so on the red line there, we have our percent inhibition. The blue dots, or the blue squares, pardon me, are the cytotoxicity. We see quite potent inhibition with the application of this drug to vero cells that have been concomitantly infected with SARS-CoV-2. The big challenge that we had with this drug was truthfully availability and knowing indication and its application to patients who would be infected with this. So the world as a whole was basically left with a lot of questions as to what drugs could work, what could be beneficial, and what was our availability. So the next step that we had taken to was looking for alternative therapeutic options. And the first place that we decided to look was, what was the host's immune response? So when we look at SARS-CoV-2 infection and severe disease, looking at peripheral blood provided a lot of insight, specifically with respect to the adaptive immune system. In the top row of boxes, you'll see several different states where we have our healthy controls, our non-ICU patients, and ICU patients labeled on the x-axis, with the y-axis being cells, cell number per microliter. And then we have our total T cells, and then broken down by subsets into CD4 and CD8 cells. The importance that we see from those groups is that as we see increased disease severity as evidenced by ICU level care, the overall quantitative level of our T cells are lower in our ICU level patients relative to both non-ICU and healthy controls. And that's concurrent with also CD4 and CD8 subsets. If we look at the bottom panel, we break it down by quote unquote disease severity in terms of mild, moderate, severe, critical, or those perished. That same trend upholds. But it wasn't just a quantitative deficit that was observed. When we look at the cells from a functional approach, specifically with the production of PD-1 expression, which is a surrogate marker for T cell exhaustion. When we look at the percentage of T cells that were producing it, broken down by either CD8s in the top boxes or CD4 in the lower boxes, or overall PD-1 mean fluorescent intensity, which will be on the right side of those, we can see that those that were in ICU level care had higher expression of PD-1 overall. So it was suggesting not just a quantitative deficit, but also a qualitative, a functional deficit, which really leads to the idea of adaptive immune therapy. This is not a new concept. It's been around for some time, specifically for viral infections in our immunocompromised patients, notably in those with bone marrow transplant. There's a lot of benefits to using viral-specific T cells. Specifically, they have targeted mechanisms of action. They can overcome issues with drug resistance when that's applicable or lack of drug availability. And there's a lot of persistence, single doses that have long-lasting effector cells. Overall, there have been two general approaches. We can selectively in vitro expand viral-specific T cells, or we can do selection of viral-specific T cells from peripheral blood of donors. So to talk about them individually, we'll look at the first approach. And one of the trials that I like to talk about the most is one that was done by Ann Lean and Helen Heslop out of Baylor, that was looking at a banked third-party CTL to treat severe viral infections in stem cell transplant recipients. What's notable about this population is, again, they were all stem cell transplant recipients, but importantly is that their inclusion criteria required persistent or refractory viral infection, either with adenovirus, CMV, or EBV. So specifically the continued detection for at least 2 weeks despite standards of care. This cohort notably did exclude patients who had pulmonary disease, which is of importance. But what they were effectively targeting was a group of highly immunocompromised individuals who were not responding to what was available. When we look at their outcomes, it's rather impressive. So if we look at the overall effect at day 42, with our x-axis measuring probability and our y-axis measuring the days post the VST infusion, nearly 75% of patients had either a partial or complete response to the viral-specific T cell therapy. but it wasn't one virus that was driving it. When they broke it down by either CMV, EBV, or adenovirus, we saw significant improvements in terms of their overall virologic response. Importantly, we're administering a viral specific or a T cell product. There's always the concern that you can induce a graft versus host type process. So in looking at the safety of these cells, what was really notable is that only 8 patients out of the cohort of 50 had developed GVHD. In those patients, 6 of them already had pre-existing or had a prior history of GVHD prior to the VST infusion, and only 2 of those patients had grade 2 or grade 3 toxicities with GVH. So this trial really highlighted 2 real important aspects. One was the power of VSTs in management of refractory or difficult to treat viral infections, but also implied a lot of safety with those viral specific T cells. Another approach is to use the selection of viral-specific T cells from peripheral blood. This is a process that we've done here at Nationwide Children's and at OSU as well. But to break it down, we identify, for an example here, for treatment of CMV, a CMV seropositive donor. We've been using haploidentical, so that being a parent or a child. If they are CMV positive, we put them through a process of leukapheresis where we take PBMCs. From those, we then incubate them with PP65. So that would be a CMV specific antigen, but you choose your virus. We can choose our own antigenic stimulation. Of those, the donor's leukapheresis product will have T cells that react with that PP65 and secrete interferon gamma. Using our Miltenyi Clinimax system, we can then do an interferon gamma capture process where we isolate those T cells and then infuse directly into the patient. This has a lot of benefits in the fact that we can condense time down to roughly about 1 week in terms of all the screening process and infusion of cells, and it doesn't require a large storage process in order to make that happen. To give an example of the power of this, we had a patient recently that was born premature at 29 weeks, and on day of life 7 developed lethargy, hypotonia, pneumonia, thrombocytopenia, and hepatitis, ultimately identified due to adenovirus. Interestingly, prior to the development of these symptoms, the mother was explaining symptoms of conjunctivitis and a little preorbital edema, which is very consistent with an adenoviral infection. Despite this detection of adenovirus, the infant progressed to respiratory failure, and we administered IV cidofovir and immunoglobulin, which are really the drugs of therapy that we have available. To highlight how significant the pulmonary involvement is, you can see on this infant who is currently intubated, there was significant bilateral interstitial infiltrate that was consistent with adenovirus pneumonitis. This diagnosis portends a very poor prognosis, specifically neonatal adenovirus infection in premature infants. The mortality rate can approach 85%. So this is where we thought out of the box and said, could a viral specific T cell product be of benefit? So we were able to enroll this patient on EIND access, and then maternally derived adenovirus VSTs were infused to the infant on day of life number 22. So to describe how this infant did, we have this graph here. On the left axis shows our adenovirus quant. On our right axis shows our rough cells per microliter. On our x-axis, we have our day of life. The solid blue line represents our adenovirus quantitative load. Our maximum detectable threshold at Nationwide is 2 million copies per ml, for which this child was at on our original diagnosis. The red dotted line is going to be that of our circulating white blood cell count, and our green line was our ALC. As mentioned, we gave IVIG and cidofovir starting on day of life 14 and administered our adeno-VST starting on day of life 22. What we saw after that was an abrupt decrease in the overall viral load, but also with that, we saw an increase in our ALC and our white count. Amazingly, when we looked at the peripheral blood of the infant at day of life 28 and thereafter, we could see HLA-A19 cells, which were specific towards mom, because this was a non-shared allele between the mother and the infant. And when we separated that out on day of life number 28, of the circulating T cells of this infant, nearly 2% were CD8 T cells and 0.6% were CD4 T cells that would have been adenovirus specific as they were derived from mom. This is a great success story because this infant survived the overall illness course and importantly represented the first case in the medical literature of a successful treatment for disseminated adenoviral infection in a neonate. It also described and gave us two important learning points. One of which is that we administered a VST product to a non-traditional group, specifically a non-immunocompromised patient. We can have a debate whether or not this child is immune compromised by its immune naivety, but overall, this was not the typical population that had received it. The other thing that was really important was that this was a patient who had severe respiratory tract disease at diagnosis. There's always been a concern that the administration of a VST product could potentially worsen that lung disease, and in this instance, it didn't. It helped ameliorate it to the point where this child was extubated roughly 3 to 4 days after we administered the T-cell product. But there are current limitations with this approach. We don't have a defined antigen yet for SARS-CoV-2 infection, and as in the case of our first point, it takes a long time for cell line generation. And one can imagine the logistical challenge in trying to use a haploidentical T-cell donor. It would require a parent or a child with prior SARS-CoV-2 infection that has resolved. So really what we need for an ideal cell-based therapy is we need something that exhibits a desired antiviral activity, a biologic plausibility for working, broad applicability, it can leverage current existing resources and rapid availability. And this is what led us to natural killer cell-based therapy. So as I hand off the discussion to Dr. Lee, he'll now go over the biologic basis, the manufacturing, and the prior history of NK cell-based therapies in immunocompromised patients.
Thanks, Chris. So about 10 years ago, we developed an approach for generating natural killer cells in the lab, primarily as a research tool, because if you can't grow them, you can't study them. And if you can't study them, nobody knows how they work. And this had been one of the limitations for a long time with studying the biology of NK cells, much less actually thinking about them as a therapeutic approach. So that approach to growing them actually led pretty quickly to thoughts around how we could use that to adoptively transfer them in a clinical setting. So what's shown in this diagram here is what ended up being our final sort of manufacturing process in a little bit of a cartoon schema, where we would send a donor to donate blood, peripheral blood, separate that as a buffy coat, which included most of the mononuclear cells, T cells, NK cells, monocytes, B cells. CD3 deplete that to get rid of the T cells, and then recursive stimulation once a week with a genetically engineered feeder cell that we developed that had all of the appropriate co-stimulatory molecules that would enable NK cells to proliferate. And what we end up with is a very highly pure, expanded and activated NK cell product that we could either infuse fresh into a patient, or more importantly, we learned that these were able to be cryopreserved into aliquots that could be thawed and infused at the bedside. So the basic structure of these NK cells is that the process of growing them also activates them, and they become a CD56 super bright hyperfunctional cell. On the left-hand side, you can see the cytokines that they produce in response to a target, showing in open circles fresh NK cells that are taken out of peripheral blood, in open squares when you grow NK cells with IL-15, and in the closed squares when you grow them with our IL-21 feeders. And you can see they have greatly enhanced production of interferon gamma, TNF alpha, and they actually begin to make IL-2, which had not previously been described in NK cells. Although NK cells broadly function as a hybrid in effector function, as both CD8 and CD4s are both cytotoxic and helper. IL-2 hadn't been described before. So on the right-hand side are a wide variety of the activating and inhibitory receptors that NK cells have on their surface, and before and after expansion. And you can see that they have greatly upregulated almost all of the important activating inhibitory receptors that let NK cells recognize and target cells that are either infected or malignant. So we translated this to a clinical trial in the context of stem cell transplant, and in particularly haplotransplant, where we gave NK cells from the same haplodonor as the stem cells were derived from. And you can see that in comparing to the historical controls, in the red line is the outcomes of haplotransplant alone, and in the blue line, the outcomes of haplotransplant with additional NK cells. And so relapse rates dropped from about 35% down to 10%. Overall survival increased from a little over 60% to almost 90%. And progression-free survival, combination between relapse and non-relapse mortalities, increased from below 60% to above 80%. And the only difference between those two lines were whether or not the patients got NK cells. Importantly, as Chris mentioned, the risk of GVH was not increased. In fact, it actually decreased in these populations because NK cells are not known to cause that as a complication. And also, somewhat surprisingly, we saw that there were decreased rates of viral reactivation, particularly BK virus and CMV. And this was perhaps something that could have been predicted, but because we were focused on the cancer outcomes, we weren't planning the study as one that would look at viral outcomes. But one of the most important normal functions of NK cells is to be first responders to viral infections. However, in the next slide, you'll see that we have moved now to a universal donor approach because manufacturing these cells for every single patient took a long time, was costly, and laborious. So it takes 3 to 4 weeks every time we found a donor to make those cells for a patient. And we had identified some key genetic factors for NK cells that would allow us to think about doing this from what we would call universal donors and identify people in the population that have ideal KIR and HLA genetics that would predict optimal function. Find those individuals, consent them to donate by apheresis, and then generate banks of cells that would be ready to infuse as a true kind of off-the-shelf product. So having established that, there are several people in my lab, particularly Brian Tullius and Mason Kararuti, as shown on the next slide, that when the coronavirus epidemic started, they were studying these expanded NK cells in the lab, and they thought, you know, we know that NK cells are first responders to viruses. Perhaps our off-the-shelf bank of cells is something that we could use rather than for cancer. Could it actually be used as an immediate off-the-shelf approach to the SARS-CoV epidemic? So on the left-hand side here, you can see there's a variety of ways that these cells interact with viruses. They recognize the stress ligands that viral infected cells have. They have direct recognition of viral antigens. When you make antibodies against a virus, they recognize that through ADCC. And then in their functional capacity, once they've recognized the virus-infected cells, they can release granzyme and perforin, FAST and TRAIL to target infected cells, and probably most importantly, release cytokines that inhibit the replication of RNA viruses and attract the rest of the adaptive immune system for a long-term response to the virus. And as Chris mentioned before, that adaptive immune response on the T cell side is also quite suppressed in these patients. So perhaps the NK cells not only having a direct effect, but would help enhance the T cell responses. So they looked in the literature, and sure enough, there was some early data coming out of China and even a few reports in the very early parts of the US epidemic that showed that the dysfunction in NK cells was quite severe. So in these patients, they had both decreased numbers of NK cells that were associated with progression of disease. The longer that they stayed in the hospital, the lower their NK cells were. There were specific phenotypes of NK cells associated with better function in when they look back at SARS-CoV-1 epidemic more than 10 years ago. And both numbers and then the interferon gamma production of NK cells is suppressed in SARS-CoV-2. So on the bottom right, you can see that the NK cells that are left, even though they're low in number, are even further suppressed in their ability to make interferon and therefore can't really help stop the coronavirus infection. So what we really had was a perfect storm of need and ability. So we had a therapeutic product that had a potential mechanism of action through production of high IL-2 that would help activate the rest of the immune system, high interferon gamma to suppress the virus. We had clinical evidence of safety in delivering these cells in transplant patients and clinical evidence for antiviral efficacy in those same transplant patients. We had cells that were immediately available. We'd been working with Be the Match Biotherapies. They have a national network of of donors that we could search to find ideal NK cell donors. We had already done that, done our first collection and expansion at the OSU GMP facility. And with the numbers of cells that we thought we needed to deliver, we could basically restore the entire NK cell component of someone's peripheral blood with a single dose and deliver more than 100 doses from a single donor, or generate 100 doses from a single donor. We had local GMP facilities that could do the manufacturing, and we had a commercial partner that had the ability to scale up that production to as much as 1,000 doses a month. So this is all wonderful if you've got an idea, but you really need good partners to do that. And so that's when we went back to Chris and said, we have an idea. How would you like to help us develop a clinical trial to test this? And so with that, I'll hand it back to Chris.
Thank you, Dean. So he really kind of hit into our hypothesis that we thought that early adoptive transfer of expanded NK cells into patients with mild or moderate disease, but at high risk of developing severe disease, could enhance patient survival, either by bringing in that antiviral effect from the NK, and then also starting to ameliorate the adaptive immune response. But this is where study design became a very challenging approach. We wanted to choose a measurable endpoint, and choosing a patient population that was already intubated on high ventilatory settings with high risk of mortality might not be an ideal population. At the same time, treating patients who were otherwise asymptomatic and doing well, there's a lot of ethical issues that run into it. So we tried to find a population that was at risk for disease progression, but targeting them early. And this is where leveraging historical controls really brought in a lot of insight. I kind of laugh a little bit at the idea of a historical control because we're talking about a few months ago. But this was a study that was published in MMWR in early April, and there are about 7,000 patients with laboratory confirmed SARS-CoV-2 infection and known clinical information, and roughly just over a third had underlying risk factors, specifically that of diabetes, heart disease, chronic lung disease, renal disease, and immune compromise. And what's interesting of those patients, roughly 44% of those with one or more conditions required admission, and of those admitted, 35% required an ICU level care. And that was wonderful for our study purposes, because what it gave us was, it gave us a target point that we could look for. So when we designed the study, we designed it inherently to look for rate of risk of disease progression requiring an ICU level care. So specifically, we would target patients with those underlying comorbidities, those that required an admission process, and then looking after infusion of cells, were we able to abate that response and then escalation of care, which is awesome in terms of looking at this from a safety profile standpoint being first. With this study too, we were able to fold in additional secondary objectives, looking at overall survival of enrolled patients, time to hospital discharge, and then exploratory analyses looking for the persistence of these expanded NK cells, and then the phenotypic immune reconstitution pre and post NK cell infusion. Overall, this led us to a study design as follows, which we already talked about the first portion of that. And once enrolled, they're able to be given a single dose, first up of 1 × 10⁷ per kilo. That, on a 50 kilogram adult, would effectively replace their entire circulating peripheral NK cell content, as previously mentioned by Dr. Lee. And then after 48 hours, we would evaluate the patient to see if they were eligible again for a second dose. Importantly, on discussions with us and with FDA, we folded in a lot of early pausing rules, early assessments, and then also ensuring that we don't have more than 2 patients at risk for the first 6, as we strongly and diligently look for safety evaluation in this, considering we've never used a cell-based therapy like this for this exact purpose. So now let's update our timeline from mid-March to where we are now. After the diligent work of a lot of individuals in the group, so that of Brian Tullis, of Maggie Lamb, of Kevin Bossi, we were able to get an IND submission to the FDA in April, as well as FDA approval in May of 2020. Now let's bring ourselves to the present date, because this is important. All those therapeutic options that we talked about that were these quote unquote magic bullets, or something that was considered exciting and possible, further studies have come to show that not to be the case. Lopinavir ritonavir did not show any clinical benefit. Hydroxychloroquine looked not only at prophylaxis, but also at treatment and randomized controlled trials, not of benefit. The one bright spot that we've had of this really has been that of remdesivir, and it is showing some promise. But like all things, there's a lot of issues that can run with it in terms of accessibility, indication or contraindications to its use, and the idea of putting all your eggs in one basket. We've seen what this outbreak has done in terms of supply chains, and having multiple options available for treatment of patients is imperative as we think about their health going forward. So where we're at right now is we're looking at site recruitment across the country, finishing up through IRB approvals, and then moving towards patient enrollment. And judging by the way things are going, even on a state level, a national level, and on a global level, I think the opportunity will present itself for us. So there's too many people to thank here. I've been a fortunate part of this collaborative effort. There's so many people in the Lee Lab who have contributed to this as well. And our thanks to them can't be enough in terms of this whole process of the FDA submission, as well as the product generation that's been done. So now to the audience, if you have any questions that you would like to have for us in the Q&A session, please feel free to put them in on the or using the panel control at the bottom. We'll now feature the 4 centers that played a critical role in the development of this invention. To begin, I get to introduce Dr. Kevin Bossi from the DDDS.
Thanks, Chris. So my name is Kevin Bossi. I'm Regulatory Operations Manager in Drug and Device Development Services. And on the next slide, I kind of give an overview of what we do. Our role here for NCH investigators, as well as OSU, is to really help guide them through the FDA process. Not just document wrangling, though there is certainly a lot of that, but to really make a regulatory strategy to ensure success, and specifically timely success as well. So we facilitate interactions with the FDA. We handle FDA meetings. We just had an early interact meeting for another program last week. We do pre-IND meetings, as well as full submissions of IND and IDE applications. We've also had a lot of success with orphan drug applications, as well as special designations like breakthrough and fast track, HUD, and HDE. So on the next slide, I would be remiss if I did not point out that our office receives support from CCTS. We're not really a core, so if you're an NCH investigator, you have full access to us and our entire team. And if you're at OSU, you can actually apply for a voucher through CCTS for our services. So obviously reach out to us if you have any questions as well. Now on the next slide, I'm showing an overview of the process of getting idea to IND, or proof of concept to actually dosing a patient. And as you can see on there, there are many steps, and there are also many potential interactions with the FDA. Each one of those steps that you take, each milestone you pass, each hurdle you jump over, that de-risks the program. So that's another thing that our team really tries to do, is that by moving things farther along, by getting down the line, by getting FDA feedback, FDA buy-in, is to really, as shown on the red line, to increase the value, the potential value to partners. Certainly an active IND is incredible in the eyes of a potential partner. So that's one of our main goals, is to really move these things forward. Now, as you see on there, we have gap analysis, interact, pre-INDs, tox studies. We kind of skipped all those on this one, and we went, as shown there, straight to the IND. We went for speed on this one, and in the next slide, I wanted to touch on sort of what we saw with the FDA on this. Now, I personally have been fortunate to work with Drs. Wallet, Lee, Lamb, and Tullius on other programs. So what we really saw here was an all-hands approach to a rapid IND creation and submission. Full IND packages are something that often can be measured in months, maybe even years. This could be measured in weeks, and we were able to get that into the FDA. Now, when you submit an IND to the FDA, they have 30 days to review it, at which point they can allow you to proceed or they can put you on hold. And what we saw in this interaction was numerous FDA interactions throughout the process of review. We had 6 clinical requests for information, I believe 5 teleconferences, and 2 manufacturing information requests. Now, we were actually able to address all of those. And that's one of the things I like to bring up is that some of those had same day deadlines, and we were able to address those. And what that really demonstrated was a careful or a calculated desire by the FDA for our success. It was clear they had concerns. As Chris just mentioned, outside of cell therapy, there's been some clinical trial failures for COVID-19, SARS-CoV-2, but they didn't want to just slap a hold on us and say, hey, come fix that later. They wanted us to succeed if we were able to meet their concerns, address their concerns. And that's one thing that we were all able to do on this together, was to be agile, to be flexible, to work with their request, and to get an active IND. So we were able to get our active IND. And with that, I will hand it back over to Chris to talk about more on the clinical side.
Thank you, Kevin. I appreciate that. So as mentioned, I'm one of the infectious disease docs and part of the host defense program. We're a group really of 5, if we want to call it that, individuals who are really devoted toward the care and management of patients with compromised immune systems. And I think to put this into a nutshell, there are so many different groups caring for a wide spectrum of immune compromised patients, all with a unique net state of immune suppression. But they do share one commonality, in that there is this overlying theme of immune dysregulation, immune dysfunction, and immune deficiency. And that lends itself to unique presentations of common and uncommon infections. And our group is tasked with the management of those infections. What this also gives us the opportunity, though, is that we're fairly heavily integrated within many of these divisions, and we get to see different approaches and different thoughts of management. And by that, we can take those lessons learned and move them elsewhere, such as, for example, cell-based therapies used largely in the BMT population and applying it to populations not historically considered to get it, as exampled earlier today with that infant with disseminated adenovirus infection. So to talk more a little bit about cellular therapy, I'm going to transfer this over to the newest member of the BMT division here at Nationwide Children's. That would be Dr. Maggie Lamb.
Thank you, Chris. So as Chris mentioned, I'm the now BMT faculty at NCH. I just completed my fellowship there, and I spent the majority of my time under the mentorship of Dr. Lee. So I'm going to be talking to you today about the cell therapy and cancer immunotherapy program here at NCH and OSU. So to understand the foundation of the CTCI program, we have to understand the nuances between traditional drug development and the development of cell-based therapies. So over the past several decades, we've really fine-tuned the traditional drug development process. And in the interest of time, I'll kind of breeze through these slides, but I wanted to just point out here that traditionally drug development is driven almost completely by pharmaceutical companies. From the discovery to drug development and early phase clinical trials, the pharmaceutical companies are really driving this process. And if we move into the next slide, we can contrast this with cell therapeutic development. And we can use the example of Kymriah here to highlight the fact that Carl June and his colleagues at UPenn drove the entire early discovery phase, lead optimization with design and validation of different CAR constructs, all of the preclinical animal models, process development in the GMP, the IND application, early phase clinical trials, and so on. And it was only after the initial clinical trials were complete that Novartis became involved in the commercialization and FDA approval of the drug. This is in contrast to the last slide I showed and really requires a research and administrative infrastructure that's not really well defined in most centers. So if we move on to the next slide, we can see that the difference here between cell therapy and drug development is that cells are living drugs. They have unique safety concerns, unique clinical trial design implication. Therapeutic drug monitoring and correlative studies for trials are vastly different than small molecules or other types of drugs. And they have really unique preclinical data expectations from the FDA for IND applications of cell and gene therapies. In addition, there's process development that has to occur in the GMP, and often these cell therapies are manufactured on a patient-by-patient basis. So there are unique feasibility concerns, often high manufacturing costs per patient, and the end product is not a uniform drug product. So for all of these reasons, when Dr. Lee came to Columbus, he had already had the experience of trying to be the inventor, the manufacturer, the pharmaceutical company all in one when he was working in NK trials and MD Anderson. So he recognized that we needed an infrastructure in place that is specific to the unique needs of cell therapy. And we're really lucky at this institution to have resources including the clinical trials offices, regulatory help, legal services for patent IP and contracts for multi-site or cooperative group studies, finance and grant project officers for budgeting and resource allocation, and finally, a GMP manufacturing space that can handle an influx of new clinical grade manufacturing, which Dean will speak about in a moment. But the core mission of the CTCI is really just to serve as the communicator between these distinct groups. And in addition to have cell therapy expertise within each area, to really help each investigator spin this complex web. If we go to the next slide, so we're here at a really exciting time where we have the potential to be the first and best in the area of cell-based therapy. We have robust NK cell research platform, a unique resource in the NK cell bank that Dean has built, a GMP facility designed to accommodate different types of cell therapies, a large patient population between both of our hospitals, and the clinical infrastructure and experience that came along with the commercial CAR T cells. And then finally, our biggest asset is really all of you and our robust untapped research base just waiting to be discovered. So with that, I just wanted to put some faces with some names. So Dr. Lee, as you've seen, is our CTCI director. Shelley Watts is really an integral part of the CTCI program as our program manager. And she does a lot of work really coordinating and organizing these different groups of people to really help our investigators get from the bench to the bedside. And she's become an expert in that area. In addition, we have, of course, Kevin, which you just met, and then our clinical research office at NCH includes some cell therapy specific coordinators, including Lori Jewell, Cassie Chancey, and I should have put Claylee Peck on there as well. And we also have our cell therapy coordinator, our clinical coordinator, Delana Sanderson. So with the help of these very specific people, we have nearly 30 cell therapy trials, either open, under FDA review, or in protocol or preclinical development at OSU and NCH. And so we're really in an exciting time at this institution. I just wanted to put our contact information up there really quickly, just in case you have any questions. Dr. Lee, Shelley Watts, or myself would be happy to field questions or inquiries about the CTCI program. And now I'll pass it back to Dr. Lee to talk a little bit more about our GMP facility.
All right, thanks, Maggie. And so lastly, we'll just spend the last 2 minutes talking about what we have for manufacturing here. So as Maggie mentioned, one of the key pieces for cell therapy is that we don't have pharmaceutical companies to rely on, and we have to take somebody's idea that they've developed and treat 25-gram mouse and make that available in something that the FDA would allow to be put into a human that's 75 kilograms. So there's some significant process development and scale up that has to happen. And so just as a little bit of humor, the next 2 slides, I kind of see this as the difference between home brew and commercial brew. I make some beer at home. My friends will drink it, but it's not really, for wide consumption. And so the next slide, if you really want to do something, you've got to build a real brewery. And so the GMP facility is kind of like the commercial brewery for cell therapy. So the next 3 slides, we'll just quickly go over the 3 GMP facilities that we have at Nationwide and at OSU. There is a relatively new viral vector core that is at Nationwide that was built specifically for generating adeno-associated viruses. And gene therapy is really in the same boat as cell therapy. We need local manufacturing in order to make it work. Next slide, there's also, we just finished refurbishing some older GMP space specifically for cell-based manufacturing at Nationwide and are just getting that up and running. And then the last slide, the longest and most experienced GMP facility is really the OSU Group under Lynn O'Donnell. They manage all of the bone marrow transplant processing for both institutions, as well as 2 clean rooms for true GMP manufacturing of cells in an ISO 7 space. And have in their leadership a good 80 years of experience in cell-based therapy manufacturing. They are the ones that have so far made the universal donor NK cell bank, and without their help over the last couple of years, we wouldn't be doing any of these studies in patients here in Columbus. So with that, I'll hand it back to Chris for some final closing comments and to move on to our Q&A.
So thank you.
I forgot, I'm actually handing it back to Bruce.
That's great.
Sorry. So Bruce Weinberg, who's the director of the CTAS program and professor of economics at OSU. Thanks, Bruce.
Thanks, I appreciate it. Lots of people contributing today. So I do want to direct everyone's attention to the Q&A function. We've got a couple of minutes that people can post questions. We have one question already that's asking about the funding details, and maybe Dr. Lee, you take that. And then also maybe if we can bring everyone back onto the screen. There was another question that for you or for Dr. Willett about how long we think that it would take before this could be available more broadly.
Sure. So for the funding question, it's from a lot of different sources. The development of the CTCI program, that Maggie discussed, as an integrated program to help people get cell therapy developed, is jointly supported by both OSU and Nationwide. The people that were on that list all have sort of dual responsibilities and funding. To specifically develop the NK cell production of the universal donor bank, we did that in partnership with Sumi Vasu at OSU, who received some institutional funds to develop that. And then moving forward for the study, we've partnered with industry that is committed to funding the clinical trial. As far as timeline, we're actually doing a couple of changes. We're going to submit to the FDA some other cell sources. So the company that we're working with is also interested in manufacturing those cells for this trial and for a larger phase 2 trial if we get to that. And we're looking for additional partners outside of Columbus so that we can get enough patients enrolled. And I'm anticipating that'll take us at least another 6 to 8 weeks to get those things in place.
Great. I was just curious personally about sort of prior experiences with commercialization, if anyone who wants to speak to that.
That's probably more in my domain. The NK cell piece to begin with was actually pretty hard to commercialize. There was a lot that was considered public domain. We went through a variety of rounds of interest. Eventually, before I came to Columbus, started a small startup company that ended up moving on to that. Since coming to Columbus, we've actually had a lot more success at commercialization through the things that we've developed around the cell therapy program. There's probably been a good 20 or so invention disclosures, and half of those have gone on to patents, that we've been able to make good relationships with industry in developing new IP and leveraging some of both of our experience and our clinical expertise in doing that. Some partnership with OSU And the clinical trials that are ongoing that we've established around Sumivasu have also had some interest in industry as well. So I think cell therapy broadly has been very exciting over the last 5 years, particularly from the T cell side. And then this year has really seen kind of finally some interest in the NK cell side.
Great. Well, why don't we leave the question open, Q&A box open, but turn things over to my colleague, Carolyn Christofouli, for some concluding remarks, and thank everyone very much for your participation.
Thank you.
Great, thank you. We're so glad that everyone was able to join us today, and we'd like to extend a special thanks to our panelists, Chris Willett, Dean Lee, Kevin Bossi, and Margaret Lamb. We encourage you to reach out to all of the panelists directly with any other questions. One of our main objectives with this series is to facilitate collaboration and an interdisciplinary network. So please feel free to reach out to panelists. In addition, we also encourage you to reach out to us about any ideas or suggestions that you've got. So reach out to Tanya Bruce or myself with other ideas for speakers and future programming. And finally, please join us next month for the August COVID-19 Inventor Showcase. We'll be featuring a research team from the Department of Mechanical and Aeronautical Engineering, Carlos Castro, Chris Lucas, Patrick Haley, and Malika Shah-Hosseini. They'll be sharing their work on a rapid COVID-19 diagnostic testing with DNA origami nanostructures. So thanks again to joining us today. Stay safe, and we hope to see you again next month.